Hello, my name is Mohammed Wahid, and I'm today representing my science project. It's about recombinant DNA, so let's get started. First of all, what is a recombinant DNA? A recombinant DNA is the general name for a piece of DNA that has been created by combining two or more fragments from different sources. Recombinant DNA is possible because DNA molecules from all organisms share the same chemical structure and differing only in the nucleotide sequence. Now we'll start off the steps of making a recombinant DNA. Step 1 is isolating genetic material. Isolation of genetic material is the process of obtaining genomic DNA or plasmid DNA in the pure form from host cell, or bacterial cell respectively. In order to achieve this, the cells are first subjected to enzymes like lysosome cellulose, or chitinase, etc. to dissolve the cell wall. The second step here is cutting DNA fragment at specific location. Restriction enzymes, also called restriction endonucleases, recognize a specific sequence of nucleotides in double-stranded DNA and cut the DNA at a specific location. They are indispensable to the isolation of genes and the construction of clone DNA molecules. And now to the third step here is joining of DNA fragments by ligation and homopolymer tailing. Homopolymer tailing is a method in which homopolymer deoxynucleotide tails are added to blunt-ended polymers in order to join a double-stranded DNA fragment to a cloning vehicle. There will be an explanation of TA cloning along with the TA sub-cloning method. And the fourth step we have here is inserting DNA into the host cell. Transduction, transformation, transfection, and injection are the four ways to achieve this purpose. But scientists must prepare for the foreign DNA by cutting it into smaller pieces, or by using restriction enzymes to cut the DNA and insert the desired portion into a bacterial plasmid before these four methods are used. And next to the fifth step, selection and screening of transformed cell. Transformed cells selected should carry at least one gene of interest, which is typified by a structural gun encoding and beneficial protein, or a gene capable of imparting a favorable characteristic to rice, in addition to a paramocin resistant gene having been transferred thereinto. And now we finish the steps. We are going to say the uses and aims of recombinant DNA. We use the recombinant DNA for medicine production, like insulin. Insulin is made of recombinant DNA, is used to produce human insulin for diabetes treatment. We also use it for vaccines. Many vaccines, such as hepatitis B vaccine, are produced using recombinant DNA technology. And lastly, we have the gene therapy. This approach involves inserting gene into a patient's cell to treat genetic disorder. And now we will talk about agriculture. Genetically modified organisms. Crops can be engineered for traits like pest resistance, herbicide tolerance, and improved nutritional content. And animal breeding is a recombinant DNA can enhance desirable traits in livestock, such as disease resistance or increased growth rates. Gene cloning. Scientists use recombinant DNA to clone genes for functional studies or to produce protein. And what is functional genomics? Is an understanding gene function and regulation through the manipulation of the. And now we have biotechnology, enzyme production. Recombinant DNA technology is used to produce enzymes for various industrial applications, such as in detergents, food processing, and biofuels. Bioremediation. Genetically modified microorganisms can be designed to degrade environmental pollutants. Diagnostics. Genetic testing. Recombinant DNA is used in tests for genetic disorders and infectious disease. PCR and RT-PCR techniques. They are techniques that rely on recombinant DNA for amplifying DNA sequence for analysis. Pharmaceuticals, monoclonal antibodies. Recombinant DNA is used to produce monoclonal antibodies for disease treatment and diagnostic purpose. And that's the end of the video. Thank you for watching.